Now that we've discussed the information models for the basal ganglia in a bit more detail, let's go back to the thalamus and see how things work at the neuron level, and then we'll see how these models are integrated with the basal forebrain. This is the information model for the basal ganglia thalamus cortex circuit. Remember that what's happening here is that outputs from the cortex recommend behaviors into the striatum. Behaviors are selected by the striatal GPI-SNR loop. Accepted behaviors are implemented by reducing the inhibition of selected thalamic targets, which increases thalamocortical projection neuron activity. This increased activity activates interneurons in the TRN, causing them to fire at the gamma band frequency. This imposes the gamma band modulation on the neurons in the thalamocortical loop, which means, for example, that the cortical neurons have greatly enhanced effects on their targets elsewhere in the cortex, or, for example, down to the brainstem to drive muscles. These enhanced effects are effectively a release to those targets, and that release is the external implementation of the selected behavior. So now let's look at how the basal forebrain plays into this, using the septal nuclei as an example. Remember, there's the competition process within CA3 and the dentate gyrus in the hippocampus to determine the appropriate cortical columns for receptive field expansion. CA3 is connected physically to CA1, and CA1 outputs drive receptive field expansions in the selected columns. While the competition is still underway, there'll be lots of activity in CA3, so CA1 will be getting lots of input. But those inputs don't reflect the final selections of columns. But the connectivity out to the cortex from CA1 is there all the time, so CA1 outputs can't be allowed to be effective until the competition process has completed. To achieve this, the septal nuclei gets lots of inputs from CA3 and a bit from CA1 and strongly targets back into those structures. The septal nuclei essentially detect the completion of the competition within the hippocampus, and at that point release the CA1 outputs to drive the expansion by placing an additional theta band modulation on top of the gamma band modulation. Let's have another look at the modulation of neuron outputs. If you were to examine the outputs of a group of cortical neurons towards their targets, in the absence of modulation, the spikes are distributed in time with no apparent temporal correlation between the outputs of different neurons. If you examine the same time interval at different times, there are roughly the same number of spikes. But now suppose a gamma band frequency modulation is imposed on the outputs of these neurons. The effect is that there's a rather higher proportion of spikes close to the peaks in the modulation signal, and rather fewer close to the minimums, even though the total number of spikes may be about the same. Now, suppose a further modulation signal is imposed, and it's a harmonic frequency of the gamma, and it has some peaks in phase with the peaks in the gamma signal. In other words, in the illustration, every third peak in the first modulation signal coincides with a peak in the second modulation signal. Actually, for gamma and theta, it's more like 1 in 5 peaks, but the principle remains, and this illustration is easier with just 3. When the two modulations are applied, most spikes are concentrated in a time slot centered on where the gamma band peaks coincide with the theta peaks, like this. And that means the effect on the target neurons will be considerably enhanced even over the gamma modulation alone. There's a very complex neuron level circuitry between the septal nuclei and the hippocampus to do this. There's 3 different types of projection neurons in the septal nuclei, for example, and it's not well understood. But the problem of keeping the appropriate harmonic and phase relationships between the 2 modulation frequencies is certainly complex. But if we go back to the circuitry that applies the gamma band frequency, the TRN in the thalamus, the basal forebrain targets the place where the gamma modulation also originates, the thalamic reticular nucleus. This targeting includes cholinergic connectivity. So at that point, the gamma and theta management systems come together to target the cortex. So that concludes the discussion of the thalamus, basal ganglia, and basal forebrain and their information models. And next, we'll move on to the amygdala and the hypothalamus.